The Hardy-Weinberg principle. It was found in 1908 by G.H. Hardy, a British mathematician, and W. Weinberg, a German physician. So they found out the mathematical relationship between allele frequencies and genotype frequencies in the Mendelian population. So this relationship is now called Hardy-Weinberg principle. It allows us to predict the population's genotype frequencies from its allelic frequencies. So now we'll consider the autosomal genes, which has two alleles, capital A and small a, in a population of sexually reproducing diploid organisms. The allelic frequencies are P and Q respectively. P for capital A, Q for small a. With random mating, the relationship between allele frequencies and genotype frequency is particularly simple, because random mating of an individual is equivalent to the random union of gametes. Conceptually, we may imagine all the gametes of the population to be present in large container. Possibility of an egg carries A is P, and probability of it carries a small a is Q. So probability of a homozygote capital A is P square, and the probability of producing small a homozygote is Q square. But the probability of it forming a capital A small a heterozygous is 2pq. So the frequency of this is, why is it 2pq? Because they say that male gamete, the capital A can come from either male gamete or a female gamete. So PQ plus PQ is 2PQ. Thus the genotype frequency expected with random mating. In this case, we'll see a Punnett square. So we take females on the above side and the males on the side. Okay, so P square, PQ, PQ, Q square. So very basic Punnett square, as you can see. So capital A, P square, capital A, 2pq, small a, q square, in sum AA plus AA plus AA. Okay, so P square plus 2pq plus q square equals 1 population. Hardy-Weinberg, this is known as Hardy-Weinberg equation. It represents the binomial expansion of P plus Q whole square. We assume that the population made at random to respect to the gene under study, that is, gametes combine randomly to produce zygotes in the next generation. If these zygotes have equal chances of surviving to the adult states, then the genotype frequencies created at the time of fertilization will be preserved. When the next generation reproduces, these frequencies will once again appear in the offspring. Thus, the random mating and no differential survival or reproduction among the members of the population. The genotype frequencies and, of course, the underlying allele frequencies persist generation after generation. This condition is referred to as Hardy-Weinberg equilibrium. When a population is in equilibrium, the genotype frequencies of such population can be calculated from the allelic frequency. The genotypic frequencies will be in proportion of P square, P2, PQ and Q square. This one of genotypic frequencies equals to 1. Okay, the genotype frequencies predicted by the Hardy-Weinberg equation can be summarized graphically. Figure will show the expected genotype frequency on the y-axis and each genotype of an equivalent value of the allele frequencies on the x-axis. So what are we seeing? What is in the y-axis? Genotype frequencies. And what is on the genotype frequencies, and on the x-axis the allele frequency. So Hardy-Weinberg expected genotype frequencies of capital A, small a, and small a genotypes, y-axis of any given value of the allele frequency on x-axis. Now that the value of the allele frequency is not graphed can be determined by q is equal to 1 minus p. So this is p, it will be decreasing, it is shown here, and the q square will be increasing, but the 2pq will peak at the middle and then will go down. So let us take a question. Problem is the frequency of the homozygous dominant genotype AA or p square in randomly mating population is 0.09. Okay, if the population in the Hardy-Weinberg equilibrium, what is the frequencies of the two other alleles? It is very simple. So we can apply Hardy-Weinberg principle to predict this. If the allele frequencies P and Q we are taking, so the three frequencies will be this, and this: P square, 2PQ, and Q square. Right, so we can apply the Hardy-Weinberg principle in this. So we are finding the P square is 0.09. So P is equal to under root of 0.09 is equal to 0.3. Right? From this we can find out Q, because P plus Q is equal to 1. So Q is equal to 1 minus 0.3. Q is equal to 0.7. Correct? Now that we have found out Q, we can find out Q square, which is equal to 0.49. Right? Now if we want to find out 2PQ, we can just do P square plus Q square plus 2PQ is equal to 1. We know the value of P square 0.09. We know the value of Q square 0.49. We don't know 2PQ that we are finding, and 1. So we'll add this. 49 plus 9, 50, 51, 52, 53, 54, 55, 56, 57, 58. 0.58 plus 2PQ is equal to 1. 2PQ is equal to 1 minus 0.58 is equal to 0.42. This is the answer. Okay, so what is the solution they are giving? So the frequency of recessive allele is this. Okay, they have found out. Yeah, we found that. We did this. They were asking other two alleles. Where's the other allele? They are only found out the recessive allele. Okay, the frequencies of the two alleles. I don't think I sometimes understand English, but this is what this is. Particular autosomal recessive disorder. There is a particular autosomal recessive disorder. Autosomal recessive disorder is present in one of the 10,000 individuals. If the frequency in Hardy-Weinberg equilibrium, what is the frequency of the two alleles A and A? Disorder of the autosomal recessive is the diseased individual phenotype AA. Okay, the population of Hardy 1 in 10,000. 1 in 10,000. This is Q square, right? Q is equal to 0.01. Frequency of AA is P square, capital A, small a is 2. Frequency of recessive homozygotes we consider Q square. So this is equal to Q square, this is equal to Q, then Q is equal to 0.01. Okay, P plus Q is equal to 1. P is equal to 1 minus 0.01 is equal to 0.99. So now we have found frequencies of, frequency of this is this, A capital A, frequency of small a is this. Very nice, good for me, good for everyone. Assumptions underlying the Hardy-Weinberg principle. So what were the assumptions underlying the Hardy-Weinberg principle? Hardy-Weinberg certain assumptions. The following major assumptions are necessary for the Hardy-Weinberg principle to hold. So number 1, random meeting. There is a random meeting within the population. In random meeting, each individual of the population has an equal opportunity of meeting with other individuals of that population. When each individual of the population does not have an equal opportunity of meeting with any other individual of the population, it is called non-random meeting. Non-random meeting. In case of non-random meeting, if members of the population choose individuals of a particular phenotype as mates more or less often than at random, the population is endangered in assortative mating. So they have made an endangered to assortative mating. For example, in human beings, there is an assortative mating for height. Short men tend to marry short women, and tall men tend to marry tall women. Assortative mating may be positive assortative mating if individuals of the similar phenotype are mating more often than at the random, and negative assortative mating if the mating occurs between individuals with dissimilar phenotypes more often than random. Non-random mating also occurs when individuals involved in mating are either more closely related genetically or more distantly related. Inbreeding is a meeting. Inbreeding is the meeting between closely related individuals, that is meeting between relatives. In human population, a meeting between relatives is often called consanguineous meeting. The mating of genetically unrelated individuals is called outbreeding. Genetically unrelated. No natural selection. All genotypes are equal in viability and fertility. This is the second condition. There is no natural selection. They are all viable, equally viable and fertile. That is no natural selection for a particular genotype. No individual has a reproductive or survival advantage over the other because of its genotype. Natural selection is a process whereby one phenotype and therefore one genotype leaves relatively more offspring than another genotype, measured both by reproduction survival. Number 3, no mutation. The effects of mutation on allele frequencies are negligible for a particular locus. If a particular locus shows a high mutation rate, then there will be a steady increase in production of mutant alleles in the population. Number 4, no migration. The effect of migration on allele frequencies are negligible. Allelic and genotypic frequency may change through the loss of addition of allele through migration of individuals from or into the population. Migration is often used interchangeably in the phrase gene flow with the phrase gene flow. Although gene flow is sometimes used to include the movement of genes into a new population founded through the colonization as well as the movement of genes into an existing population. To a population geneticist, the word migration means the movement of organism from the population in which it was born to another in which it reproduces. If an organism returns to the place where it was born to reproduce, to a population genesis, there has no migration. So weird. For a genesis, if an organism goes to different organisms and reproduce in that different area, so this is a migration. But if this organism goes there, comes back, and now reproduces, now there is no migration. This is what it is saying. Number fifth, the fifth condition is large population size. Population is sufficiently large so that the frequencies of allele do not change from one generation to the next generation because of chance. In small populations, significant random fluctuations in allele frequencies are due to genetic drift. The random change in allele frequencies in a population by chance alone due to sampling error from one generation to next generation is called genetic drift. During drift ultimately leads to fixation of one allele at a locus and loss of all other alleles. While it occurs in all populations, genetic drift can be a major driving force for changing allele frequencies in small populations. The smaller the population, the more extreme it tends to be. If these assumptions are more or less met, then a population is expected to be in Hardy-Weinberg equilibrium. In this situation, two results are expected. First, the frequencies of the alleles will not change from one generation to another. Second, the genetic frequencies will be in proportions p square to pq and q square. When the genotype are in these proportions, the population is said to be in Hardy-Weinberg equilibrium. In reality, no population satisfies the Hardy-Weinberg equilibrium completely. Nevertheless, in large natural populations with little migration and negligible natural selection, the Hardy-Weinberg equilibrium may be nearly approximated for certain genes. Thus, Hardy-Weinberg principle has two aspects. In an infinitely large randomly meeting population free from mutation, migration and natural selection. Number 1. The allelic frequency in a population will not change from one generation to the next. Allelic frequency equilibrium. Number 2. The genotypic frequencies of the population are determined in a predictable way by the allelic frequencies. Genotypic frequency equilibrium. If the allele frequencies in a population with two alleles at a locus are p and q, then the expected genotype frequencies are p square, 2pq and q square. The frequency distribution will not change from generation to generation once a population is in a Hardy-Weinberg equilibrium. What is the validity of the Hardy-Weinberg equilibrium, we think? So it is possible to establish whether a population is in Hardy-Weinberg equilibrium for a particular trait. Always remember, only one particular trait we are talking about, always. If all possible genotypes can be identified. Consider a system with two alleles A and B with three resulting genotypes. Capital A, capital A, capital A, small a, small a, small a amongst 1,000 individuals selected at random. The following genotype distribution are 800, 185 and 15. From this data, frequency of allele capital A, only P, is, how are we finding this out? what are we doing here? 2 times 800 plus 185. About 2000. Why are we doing this? Let us think. Okay, so we have to find for A only. So A plus 2AA plus AA. So they are taking this like A plus 2 A's from here. Okay, and for small a, 2 small a's plus A. Okay, so the small a's, we are dividing this, right? So it will be half of 185. Now this half of 185 will be multiplied by this 2, and this is A plus A, 800 twice the 800. Okay, so this is 2 times the 800 plus 185. So now if we are trying to add these up, so 800 2 times 185, okay, and the 15 and 15, so it will be the double of the 1000 individuals. It will be 2000. So this 2000 will be divided by this whole number, and we will be getting 0.8925. This is what we are doing here. Similarly, here this 185 is from this, half of this is multiplied by 2, and the twice of the 15s divided by 2000, we are getting this. Now consider that the expected genotype frequency would be if the population is in Hardy-Weinberg equilibrium, and compare these with the observed values. So we found out the capital A observed was 800, okay, but the expected was this much, similarly others. These observed and expected values correspond closely, and formal statistical analysis chi-square test would confirm that the observed values do not differ significantly from those expected values of the population is in equilibrium. Now take a different system with alleles capital B and small b amongst 1000 randomly selected individuals. So 430, 540 and 30. We are finding its B. So similarly, twice of the capitals, and take one whole of 540 and divided by 2000, we are getting this 7. B or P is 0.7, small b or Q is equal to 0.3, so 0.7 plus 0.3 is equal to 1. We found this out, but the expected values were supposed to be what? P square. This BB is supposed to be P square is equal to 0.7 square is equal to 0.49 into, why we are multiplying it by 1000, because there are 1000 individuals, so it will be 1, 2 and 3, 490. This is the expected value, okay, but we have observed is this. It is also significantly closer. The first value is significantly closer, but we find that this value or this value are very different. They differ considerably, with an increased number in heterozygotes at the expense of homozygotes. The heterozygotes are lot more than the homozygotes. This deviation from the RD-Weinberg equilibrium may be due to increased numbers or the heterozygotes, such as heterozygote advantage and negative associative mating, that is attraction of the opposites. Now we are doing the extension of Hardy-Weinberg principle equilibrium. Extension of Hardy-Weinberg equilibrium, it can be extended to multiple alleles of an autosomal gene, sex-linked gene, and polyploidy. So we'll first be seeing for multiple alleles. Hardy-Weinberg principle can be extended to a gene with multiple alleles. Okay, one gene, multiple alleles. Calculation of the allelic frequency with multiple alleles employed the same rule that we used with two alleles. Suppose in a population of the diploid organism, an autosomal gene has three alleles, A1, A2, and A3, with the dominance hierarchy. A1 is more than A2, that is more than A3. Let P, Q, and R represent the frequencies of A1, A2, and A3 respectively. In this case, the allelic frequency will be, so we are finding P, Q, and R. So 2 times the total worth of A1s plus A1 and A2, and then A1 and A3. We are finding this for P. Okay, and that twice the total number of individuals. Similarly for Q, twice A2 plus A1 A2 plus A2 A3, and for R, twice A3 A1 A3 A1 A2 plus A3, and divided by the total twice the total number of individuals. With three alleles, the allele frequency of an all allele must be equal to 1. So P plus Q plus R is equal to 1. To calculate the expected genotype frequency if the population is in Hardy-Weinberg equilibrium, we use trinomial equation. So the trinomial equation, P plus Q plus R's whole square is equal to P square plus Q square plus R square plus 2PQ plus 2PR plus 2QR equals to 1. The homozygotes will occur with frequency P square, Q square, R square, and heterozygotes will occur with frequency this, and this. Problem: in a large randomly mating human population, the frequencies of IA, IB, and I, the alleles for blood type, is 0.7, 0.2, and 0.1 respectively. Calculate the expected frequencies of each blood type. So solution is, in this population in the Hardy-Weinberg equilibrium, if this is in a Hardy-Weinberg equilibrium, the genotype frequencies can be calculated as this equation. Okay, so we now know P is equal, we have given is P is equal to 0.7, Q is equal to 0.2, and R is equal to 0.1. So P square is equal to 0.49, Q square is equal to 0.04, and R square is equal to 0.01. Satisfied? Right? Let us calculate the expected frequency of each blood type. So what are the blood types? A, B, O. How we will be getting A? IA, IO, IB, IO, and this is only OO. And the fourth blood type, AB, IA, IB. Right? So this is what? AA, BB, OO. Okay, and now the heterozygotes, 2PQ, 2QR, 2PR, and they are P, A, Q, B. Q is P, R is O. So this is P. P is A and R is O. So this is A. Correct. Now let us find out. So this is twice P 0.7 times Q 0.2. We will not be taking the 2. 0.14. Now this is 0.2 times 0.1. भाई 2 से मल्टीप्लाई करना चाहिए ना, तभी तो हमारा गणित बैठेगा. आई थिंक यहाँ पे 2 आना चाहिए, यहाँ पे भी 2 आना चाहिए. 0.28 is equal to 0.04, 0.04. Okay, and this is twice P is 0.7, R is 0.1 is equal to 0.14. Okay, so for blood type A, we have (0.49) plus 0.14 is equal to 9, 10, 11, 12, 13, 4, 5, 6. Okay, for B we have 0.04 plus 0.4 is equal to 0.4. Okay, we are multiplying it 0.12. Okay, for 0.08. Okay, for O we have 0.01 plus no other, that means it will be 0.01. and AB we have no here but here 0.28. Done. Next question is sex-linked genes. The sex-linked genes are located on the sex chromosome and the traits are expressed by these genes are termed sex-linked traits. This term are generally referred to X-linked traits that are expressed by genes on the X chromosomes. The Hardy-Weinberg principle can be extended to the X-linked genes also. If a gene is sex-linked, the heterogametic sex, the human male, has one copy of the gene and termed as hemizygous, while the homogametic female 2 copies. Equilibrium, the frequencies at the equilibrium of P and Q, the heterogametic sex, but P square 2PQ and Q square for the homogametic sex. For males we are taking P and Q equilibrium, but for females we are taking this equilibrium. That is, for heterozygote sex, the genotype frequency is equal to the allele frequency. The genes are equal to allele frequency. But for X-linked recessive allele A, in nature, its population is Q and dominant is capital P is P. genotype we are seeing in males. If males have X capital A, so it will be P. X capital small a, it will be Q. Simple. Okay, we discuss capital A, okay, P square, capital A small a, 2PQ, small a, Q square. Example, suppose the recessive allele of X-linked recessive disease in human population, small a, is 0.02. Frequency for males, if this is Q is 0.02, then P is equal to 1 minus 0.02 is equal to 0.98. Correct? Calculate the proportion of diseased individuals in the population. Diseased. Since the males need only one copy of allele is 0.00, females must inherit 2 copies of the alleles. That is homozygous to be disease. So females they require A square or AA or Q square to be diseased. So it is equal to 0.02 whole square is equal to 0.004. If we assume half the population of male and half the population of the female, frequency of the, if we assume that half the population of the male and half female, then overall frequency of the diseased individual is half of 0.0 plus half of 0.0004 is equal to 0.0102. Now we'll think about polyploidy. What happens when polyploidy is occurring? More than one allele is possible. The hierarchy generalized in polyploidy organisms that two sets of chromosomes. In case of diploid organisms with 2 alleles, the binomial is this. 2 alleles, so we have this. When we have n alleles, we'll have polynomial expansion. So if we have this 4, n is equal to 4, then a will be 4, p raised to power 4. For this 3 is capital A, 3, 1 small a, 1, and we are multiplying it by 4. 2 capital A's, 2 small a's multiplied by 6. 1 capital A, 3 small a multiplied by 4. This is a male expansion. So this ends with the Hardy-Weinberg principle. We can do an exercise question. Okay, let me just add a page down there. So the question is, in a population, the frequency of a phenotype is 16%. A. What is the frequency of recessive allele? B. What is the frequency of heterozygous individual? Let us see the answer. We have the frequency of recessive phenotype, that is 16% or 0.16, that is equal to q square. We have q square. We can find q from this. So this is the frequency of allele. Now, for everyone who knows, P plus Q is equal to 1. So P is equal to 1 minus Q. 1 minus 0.4 is equal to 0.6. So P is equal to 0.6. Frequency of dominant allele. We found this. But we have to find frequency of heterozygous individuals. Now we need to find out the heterozygous individuals. So we know P plus Q is equal to 1. So P plus Q whole square is equal to the 1's whole square. LHS is equal to RHS. If we are squaring the LHS, we are squaring the RHS. So the product of this square is P square plus Q square plus 2PQ. That is what we wanted to find out. This. So we have P square. What do we know about P square? 0.6's whole square plus Q's whole square 0.4's whole square plus 2PQ 1. 0.36 plus 0.16 plus 0.2PQ is equal to 1. 2PQ is equal to 1 minus 0.36 minus 0.16 is equal to 1 minus 0.52 is equal to 0.48. 2PQ. C of heterozygous individual. So we did the whole Hardy-Weinberg principle from Pathfinders. What did we do today? What is a Hardy-Weinberg principle? What are its conditions? The graph related to the Hardy-Weinberg equilibrium. What are the assumptions? Number 1, there is random mating. Number 2, there is no natural selection. Number 3, there is no mutation, no migration, and there is a large population size. So we saw the validity of Hardy-Weinberg principle. We saw the extension of Hardy-Weinberg principle, how multiple alleles can be governed under this equilibrium. We did questions for it. We saw the 6-linked genes, and finally the polyploidy. Polyploidy is, if there is a question, it will always be solved by the binomial expansion. Always remember this. So the thing is, why is Hardy-Weinberg principle so cool? Because whatever the LHS is, p plus q, it is always equal to 1. So if it is p plus q squared or thrice or quarter, like 4 raised to power 4 times or n times, when the RHS is raised to power that times, it will always be 1. This is not changing. Thank you. Have a nice day. Have a nice 24 hours. Goodbye. Tata, toodles.